you look at these pictures, here's a peritrichus. Here's a Proteus vulgaris, in fact. So you can see why that's a swarming organism, because it's very, very, flagellated. It's very modal. It has all those flagella. Here's a Pseudomonas that has just, it's monotrichous, it has one, but it's a polar flagella because it's just an end like that. And here's a Locotrichus, where you have a cluster at one end, but you could have something that's Locotrichus that would have a cluster at either end. Okay, so those terms you do need to know. Amphitrichus has it at both ends, but polar, you could have one or both ends. But it's at the end.
How do you know the difference between the polar one and the flagella?
If you said it was polar, you wouldn't really know if it was for sure at one. If it was monotrichous and it was a polar, then you would know there was one. And if it was, what was the other one that you asked about? If it's amphitrichus, then you know it's a polar. Like you think of like ambidextrous, right? Like when people are both left-handed and right-handed, that's how I would remember that one. Polar just really just means the end. Did that answer? It's kind of like they could like overlap a little bit. So again, filament, hook, basal body, I don't need you to, draw that out or anything like that. There's some slides that show how, what you should know about the archaeal though is thinner flagella, and they are different. So like everything else is different about them too. So you just know that they're composed of more type of different protein. They're not hollow. They look different. They hook up differently, but mostly you can see the structure is different. The thinner, different structure, that would probably be what you would need to know for the archaeal. All of this stuff about motility, I want you to see this, but I'm not going to ask you about the different ways in which they are modal. I just want you to know that it's known and understood. I mean, why they're modal, I think, is pretty straightforward. Why anything would be modal, because it allows it to really adjust where in the environment it is. Even when things don't have a flagella, they can be modal in other ways, right? They can glide on like a slime layer. Also, things like fimbriae can cause sort of a twitching type of movement too. So things can move around. Sometimes they just move around haphazardly in their environment. But if they do have flagella and can move easily, they're going to regulate where they are in their environment, like move towards nutrients, move away from anything that's hazardous, move into different areas of oxygen or osmotic pressure and such. So when we look at this, how they move with the flagella, they actually, can see how they move with different, a tumble, a forward run, or so, how those kind of interest, but I'm not going to ask you to memorize exactly how they're doing this. Spirochetes, remember when we talked about the shape of bacteria, and I said spirochetes are something really that are actually different and unique. They are spiral shaped. But spirochetes, again, we'll talk about them at another time, but here's one more piece of information about spirochetes. They have a different type of flagella that's called an axial filament, or a periplasmic flagella. So this axial fibril or axial filament, and they're also sometimes referred to as periplasmic flagella because this filament is found in the periplasmic space. So if you look here, it kind of twines around the particular organism. So you see this axial filament, spiral flagella, periplasmic flagella. It's all referring to that specific type. It's only found in spirochetes. Spirochetes are flexible, spiral-shaped bacteria that just have other unique qualities to them. They're going to be different than just sort of the typical spiral-shaped organism. So I mentioned about the twitching and gliding. The twitching, you can have that from pili or fimbriae. They can glide on that kind of slimy layer. There are bacteria that produce slime layers that are something even different than what we talked about when we talked about the structure of the slime layer. There are different bacteria that actually produce a slimy substance in which they will move around on, and it'll actually even leave a trail that looks like how a snail leaves a trail. When we really start talking about other bacteria besides the ones that are just like rods and cocci and such, there's a lot of really unique types of bacteria. And then the last thing that I'm going to talk about is the bacterial endospore. We know a lot about endospores, but what you need to understand about endospores is going to be the structure of the endospore. So right now, because we work with spore staining, because we did a lab in which we tried to look at some of the resistant qualities of the endospore, but since there were no endospores, we didn't really get much of a result. But you got to see endospores, so you know a little bit about that. They're resistant to a lot of environmental conditions: heat, radiation, chemicals, desiccation. Okay, so they can survive. They found bacterial endospores, like basically encased in amber that, could still be viable. They found them in mummies. They found them, hundreds of years old that are still viable, thousands of years old. Where they might be in the cell before it's become a free spore, that is characteristic with certain bacteria. So certain ones tend to form the spore in more of a subterminal area versus a terminal versus a central area. But what I want you to know then is this structure. So usually I could either list out the name or ask you to diagram the layers of an endospore. And this is kind of giving you the information really more from the outside. You have an exosporium on the outside. Under that, the spore coat. So when we did the spore staining, we were trying to get the spore coat to pick up the actual stain, even though on the outside of that is actually something like exosporium. That's a thin covering. Under that, you have a cortex, and then you have a core. And this just is kind of showing you that basically inside the core, that's where you find the nucleoid and ribosomes. The cortex has peptidoglycan in it. Then you have the spore coat and exosporium. So if you look at this, this kind of shows what it looks like, looking at different type of microscopy. But here, this core has the nucleoid inside. Then you have the cortex. Then you're going to have the spore coat, and then the exosporium. So this kind of add a few extra things in here because it's showing you the outer coat and inner coat, just because it's kind of showing like the depth of that. I thought there was no. So you kind of have to use, probably this list makes it a little bit easier to follow. Yes, so yes.
So in that image that's like the screen, you have the diagram for you.
Yeah, and but stick to this, like where you have the exosporium, and then you have the spore coat, then you have the cortex, and then you have the core. You can do it like that, rather than try to get it. Like here, they're showing you the nucleoid, but that's inside the core. And then the cortex, and then the outer and inner coat, but then spore so resistant? This also is controversial because there are other structures associated with bacteria that are also very resistant that don't have some of these same components. So obviously the fact that there's a thick coat, that is going to be something that makes it seem obvious, oh yes, that's going to make it a little bit resistant. But it also is thought to be resistant because of this calcium and the dipicolinic acid, sometimes referred to as calcium dipicolinate. However, if you look at some of the other structures that are also very resistant associated with bacteria, they don't have calcium and dipicolinic acid, which is really referred to as DPA, dipicolinic acid. So calcium is something that can lead to some heat resistance. And so they do know that this probably is a factor. They just are not really sure how much of a factor it is. There are these small acid-soluble DNA binding proteins, referred to as SASPs. It's dehydrated, so you have a low water activity. So that means if it's dehydrated and exposed to heat, you're not going to have like the kind of reactivity that you have if there's a lot of moisture there, which then can be destructive. And then you have that outer spore coat and exosporium. So again, these are, somewhat believed to be part of it, but is really not felt that it's truly understood exactly why they are as resistant as they are. And mostly because you don't see these same factors in the other resistant structures like exospores and some of the others. If you look at the process of sporulation, you can see here too, it occurs in some hours, maybe even up to 10 hours. Maybe that's something that, has caused a problem when we're making, the spore suspensions here, even though, Jan has made them early enough that they still shouldn't form spores. But if you look at this process, again, I'm not going to ask you to memorize this process, but I want you just to be aware that it is really understood. You can see here's like a good representation of endospore. You see the exosporium, the spore coat, the cortex, and the core. But it just kind of shows, how it kind of is starting to invaginate a little bit, break up the genetic material a little bit, and then start to surround the cell, and then finally the cell kind of just disintegrates and you end up with a free spore. When it germinates, that's when it comes back out into that vegetative cell. And usually something happens like the type of environment that will cause a chemical reaction that will then allow the spore to start to open up and then allow the nutrients and moisture to get in, and then it germinates. And you have one cell becoming one spore, becoming one cell again. So there's no kind of replication. So that is the bacterial cell. Everything you wanted to know about a bacterial cell. But before we leave, I do want to show you what you need to know from Chapter 6. Chapter 6 is one of those ones that's listed on your paper that you have to look at on your own, and you'll see why. It's actually helpful for you to go through Chapter 6 because it gives you a lot of the information that you need to know about what you're doing in lab. Chapter 6 is just about nutrition, and if you look through here, it really just lists these macronutrients, micronutrients, the same things that you learned about when you learned about eukaryotic cells. I will ask you on the exam to name some of the essential nutrients that bacteria need to grow. That's not something that I need to lecture on. You just look and you see that you need the carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus. You learned about these things, right? Potassium, calcium, magnesium, iron. Does that sound familiar? I'm not gonna ask you like multiple choice and true false questions about this, but just simply that you can like show me that yes, you remember that bacterial or prokaryotic cells need the same types of nutrients that eukaryotic cells need. Okay, macronutrients, micronutrients. Then here's this terminology again that you already learned about, heterotrophs, autotrophs. And then remember how we learned about like photoautotrophs and chemoheterotrophs, and how I said with bacteria that you could have, you know, all different combinations of those, and then lithotrophs versus organotrophs. So here it's kind of listed separately. Remember, you already learned about this, though, the electron source. And typically, if you look at regular eukaryotic cells, you have something that is a photo, litho, autotroph, or you have a chemo, organo, heterotroph, because one was inorganic carbon and something that was photosynthetic, and the other was organic carbon, and the breakdown of those carbon sources. But with bacteria, they can be kind of all mixed up. So you want to just, you know, reacquaint yourself with those terms. Photo, organo, heterotrophs, chemo, litho, autotrophs. Just because there's that much diversity that you see in the bacteria. You know what I'm talking about, right? Because you have things that are photosynthetic that still use organic compounds and things that use inorganic compounds. Am I losing you guys? I know you know this. You just have to remind yourself again. So see, all of this stuff here, you don't have to know all of this stuff. You just have to be aware of the terminology and those micro and macronutrients. And then it kind of goes into culture media, obviously, because the two go hand in hand. And in the culture media, they talk about things like we already learned about that in lab, the defined versus synthetic, defined versus complex. Remember that? Defined where it had all the ingredients like in specific amounts, and complex were things like beef broth and components, blood in blood agar, things that have multiple different types of nutrients, but we don't know exactly how much they have of any one thing. And then it goes into enriched, selective, differential, you know, supportive things. So you've already had to learn about selective media and differential medias. Enriched medias are just those that have nutrients added to encourage the growth of specific organisms. Supportive medias are those like tryptic soy agar or nutrient agar or broth that just have ingredients for general growth. So here you can see that tryptic soy agar, enriched medias, selected medias, McConkey and EMD. So this is stuff that you know, differential. Isolation of pure cultures, this is what you know already. Streak plating, spread plates and pour plates, dilutions. And then this is kind of a little bit of what you guys had to do last time. So really this chapter doesn't really give you a whole lot of new information. What I would be expecting you to know from this chapter would be those micro and macronutrients, and also those terminologies. Okay, the rest of it is actually more associated with lab, but I would expect that you know what a differential media is, what an enriched media is, what a selective media is. So I could ask you a question on something like that. I'm not gonna really ask you about culture characteristics or, you know, the pure cultures or street plating or dilutions or anything, because we really covered that really adequately in lab. And when it comes to things like enriched media, we're not really using an enriched media. We are gonna have a demo with Blood Auger, I should take that out. But other than that, probably most of that is gonna come out on a quiz, so I'm not necessarily gonna need to really test you on that in lecture. But did you guys you got the code, right, for signing up with that Learn Smart or that Course Smart? Remember on the first exam, I took questions directly out of that, and I'll do that again. So I'll at least include, you know, a certain, like at least a few questions for each one of the chapters directly out of that. I mean, really a lot of the questions are the kind of questions I would ask you anyway. But I also got a code for for the 9th edition of Prescott, which I don't know if maybe you're the only one that has it. Does anybody else have the next edition? You guys all have the 8th edition, right? You can still use the 9th edition if you want to go in and use some of those chapters. You just have to like look at the chapter and see what it's called. But the book is very similar. So if anybody wants that, I'll try to get it out. Like, he emailed it to me last night, but then when I looked at it today, like, I couldn't find where the code was. It's like the code is below and then it wasn't there. So I emailed him back and then he apologized and supposedly sent it again. And when I looked again, I didn't see it. So when we're in lab, I'll try looking it up on the computer. But all the stuff for exam 2 is, you know, under exam 2 on that Course Smart thing. And I don't know if it helped you guys. It did help you. I mean, it's just another way it forces you to kind of look at the stuff. And the Learn Smart thing that goes with the next edition, they don't have it with the 8th edition, but that goes with the next edition. It just asks you a whole bunch of questions so that you're just kind of going through it. It's a lot quicker and it's not something that you turn into me or anything like that. So you can still like go to the chapter on nutrition or go to the chapter on the bacterial cell, and it's still going to ask you, you know, obviously related kind of questions. So make sure like over the break that you guys are, you know, catching up with all that, because basically all we have to do to talk about on Tuesday when you come back is the chapter on growth. And the chapter on growth is not very much either. If you look through the chapter on growth, part of it is about cycrophiles and neurophiles and halophiles and all of those terms that you also learn about in lab. But what we really have to learn about for the exam would be the growth curve. So if you're trying to be prepared ahead of time, you'll read the part about the growth curve, because that will be one thing that on the exam I will be asking you to write out in detail, and I will talk to you about it in detail. But again, if your study time is limited going from Tuesday to Thursday, that would be something that you would want to read a little bit about before, because it's pretty simple, but there is still a good, you know, amount of information. When we talk about the growth curve, it's things like the log growth phase, the stationary phase, the log death